So we are covering this histology of these female reproductive and the male reproductive for the day, and we cover this particular content first. It's kind of like a complex content which we have in the histology on the ovary, which is the female reproductive organ. That is a part of reproductive organ and is a part of endocrine tissue as well. So it secretes some hormones like a progesterone and estrogen, and apart from that it produces the gametes, which you call them as ovum in case of the reproductions, which plays the role for the fertilization. So the gross part we already have known. So we are just like going through this particular histology part. Now the covering this particular like a tissue or this ovary having a layer of the cell, we call them as epithelium layers. And this particular epithelium, because it is related to the germs or germ cells, we call them a germinal epithelium. So this is a single layer, flat to cuboidal layer, which is covering the entire ovary, and it is present on the outer side to it. Then deep to this particular germinal epithelium, we have a connective tissue layer, we call them as Tunica Albuginea, which is very thin layer, and then we have this particular ovary tissue. Now ovary tissue is split into two parts, we call them as cortex outer side and medulla inner side. The medulla is filled up with the blood vessels, lymphatics, nerves, etc., etc. The cortex is covered with the ovum, which is covered or layered by cells which is present around it. Ovary, which having got this architecture, we call them as ovary and follicles. That means the ovum, that means the follicles which is present along with that, which contains two things. One is germ cell, which we call them as oocyte, and surrounding the germ cell we have a tissues or which have the cells which is surrounding to it. Together we call them as ovarian follicles. So we have this germ cell which is coming from this yolk sac, migrating into it and growing into it, and they have this resident cells which is present to it, which is the cells which is present in it, we call them as follicular cells, which is covering around these germ cells. Together we call them as ovarian follicles. Now these ovarian follicles are of different stage and different looks. So they are of different size, shape and looks, and that's why we call these follicles in different names. We call them as primordial follicle, primary follicle, secondary follicle, Graafian follicle, dead follicles we call them as, which is known as corpus luteum. So these are the different stages of the like follicles can be seen within this particular like this whole ovarian tissues, just like the Big Bang theory. So once there was a big blast which is going on far away, and we can just like if I move back, we can trace back to that light year substance, we can still see the Big Bang going on and till the cell till the planets are growing like that. So we have the different stages of ovarian follicles. So starting from the tiny one to that dead one which is visible, and they are present around the cortex, like outer side of the ovary. The middle part which is occupied by these blood vessels, lymphatics, nerves, etc., etc. So if you see this particular ovary is coming into these two content, cortex and medulla. The cortex outer, medulla is middle or inner side. Covering to it, we have a connective tissue layer, we call them as Albuginea, Tunica Albuginea. Other side or outer side to it, we call them as Tunica vaginalis, outside to it. Then deep inside, we have this Tunica intima, which is present with this vascular layer, we call them as Tunica vasculosa. And the most outside, we have a single layer of the cuboidal cells, flats, we call them as germinal epithelium, which is covering this entire thing. Now this point of concern is that this particular follicles. Now follicles contains two things. One is the cell, which we call them as oocytes. Now oocytes are, which is going to be discussed here, are of two types. We call them as primary oocyte and secondary oocyte. Now what happens with this oocyte? We know that this particular oocyte that migrates from the germ cell and settle there. This particular oocyte are arrested at a phase. We call them as arrested in the first mitotic division. So the diploiding stage, it is arrested there, it remains there. We call this oocyte as primary oocyte. Now it remains there till the puberty phase. So once they start maturing, once it start coming and settle down over there, it comes as or stays as the primary oocyte, which is arrested at the mitotic division second diploiding stage, and it remains there till it reaches to the pubertal age. Pubertal age means we have the hormone influx coming in at a particular age. When this particular mobilization of the oocyte keeps on happening, that particular oocyte after stimulation reaching to the pubertal age starts to continue that particular cellular division and completes this fast meiotic division, and then it splits into secondary oocyte and polar body, which is exited out, and the polar body and the secondary oocyte goes into meiotic divisions or duplication part. We call them as second divisions, which is only possible completely if fertilization takes place. If fertilization does not take place, this particular secondary oocyte that is liberated, that will be arrested at metaphase, and it will remain arrested at metaphase, and it will be absorbed or destroyed unless until it meets with the sperm. Only this particular secondary oocyte, if it is meeting with the sperm, the continuation of meiotic division, which is arrested at the metaphase, will be continued as this continuation phase, and the second polar body will be exited afterwards. So, these are the physiological part or like pretext part of this particular ovum or the oocyte that we have decided or that we have discussed. Now, this is how this particular whole thing schematically can be described. So, it is kind of like spreaded across, and this has got like a cortex and the medulla, the central part medulla, outer part is cortex, and this particular things which you are seeing here different stage in different stages of follicles. So, starting from the tiny one to this little bit bigger one to this particular this intermediate one to this mature one or to this dead one, which is the after expulsions of the ovulation that happens. So, this started with this covering with this particular cellular layer, single layer, we call them the germinal epithelium. Then this germinal epithelium, then we have the tiny tissue, white tissue, we call them as Tunica Albuginea, very thin layer, and then we have this particular cortex present in it, and the medulla is free from this particular like a follicles, we call them as like medulla contains this particular blood vessel lymphatics. So, this together we call them as follicles, that means the germ cells surrounded by a layer of flat cells or different looking cells, pubertal cells, which is holding in surrounding this particular oocyte, we call them as follicles. Now, these follicles can be classified into several types. We call them as primordial follicles. This is the tiniest form that comes in these germ cells, and they are surrounded by a flat epithelium cells. So, this is the oocyte, primary oocyte that comes in here, and it is neighbored by a layer of cells, which you can see this one where I am placing the cursor. So, these are the flat cells which is surrounding this, and together this 1 and 2 becomes follicle, and this follicle is the tiniest one, we call them as primordial follicle. So, this primordial follicle is having this particular oocyte or primary oocyte, as you can see, is tiny one and surrounded by cells, we call them as follicular cells, right. How do the follicular cells looks like? Skinny, flat, does not have much of girth, flat follicular cells. So, this is one we call them as primordial follicle. Now, this primordial follicle with nutrition and everything, it grows a little bigger, that becomes primary follicle. So, from primary it becomes primordial follicle. What happens to the primary follicles? The oocyte becomes little bit bigger, and this particular follicular cells becomes little bit bigger as well. So, this particular bigger follicular cells, these were skinny one, you see that tiny one, flat, becomes cuboidal cells, becomes little bit bigger. So, this particular follicular cells which is surrounding in here becomes single layer. Now, this particular follicular cells becomes multi-layer. That means, this is what we are seeing here, a primary phase of the follicular layer. So, this particular follicular cells which is single layer, now it becomes stratified, that means it grows in numbers or in layers. So, that particular single follicular layer becomes multi-follicular layer, clear? That this multi-follicular cells, as it is becoming multi-layer, we change the name from the follicular layers. The inner layers of the follicles which is surrounding this, we call them as granulosa cells. We call them as follicular cells, the outer one, then we call them as granulosa cells which are inner side of it. So, if it is another layer of cells present in here of the follicular cells. Now we do not call these as follicular cells. Now we call them as inner layer as granulosa cells, and this one is outer follicular layer. It remains there. Now these granulosa cells becomes further divided into and becomes more and more multi-layered. So that particular granulosa cells, again it becomes granular multi-layers, then again it differentiates into two sublayers. So this particular granulosa cells, as it becomes multi-layered, we call them as theca cells. Now these theca cells are of two types, one is inside, other is outside. We call them as theca interna and theca externa. So same follicular cells that becomes granulosa cells, that same granulosa cells becomes theca cells, and theca cells become external theca and internal theca. The internal theca is vascular, the external theca is avascular, less vascular. Now this internal theca cells becomes the hormone producing cells eventually, what secretes the progesterone, estrogen eventually, right. Now what happens after this? This inner cell layer of this granulosa cell that becomes theca interna, that secretes a glycoprotein substance. What do you mean by glycoprotein substance? That is mixed up, a chemical substance which is mixed up with the protein and as well as glucose tangled together. We call them as GP layer, glycoprotein. So this theca interna cell secretes or produces some kind of like a GP layer which surrounds this particular developing oocyte. So that particular layer, GP layer, which surrounds this particular oocyte, we call them as zona pellucida. So what happens in this particular primary follicular layer? We have this oocyte, primary oocyte arrested at diplotin stage, then having a ring which we call them as GP ring, zona pellucida. Then we have a layer of cells, we call them as theca interna, then outside theca externa, then outside theca follicular cells. So this is how this whole network is arrested. So this becomes the fully matured primary follicular cells, and at this stage it remains till puberty. Say for example, the individual is born with 2 lakhs of this particular primordial follicle, and that primary follicles becomes fully mature primary follicle at the pubertal age, say for example 1 lakh. So from 1 million to some number, it becomes 1 lakh matured follicular cells. That is ready follicular cells before puberty. Now as the puberty gets in, we get the hormonal influx which stimulates this particular cells provincially, individually, tags in, and this particular primary follicular stage keeps on growing for secondary follicular stage, which goes in. What happens in the secondary follicular stage? Now it creates a space within this theca cell layer, theca internal layer, or this particular granulosa cell layer. Within this, it creates a cavity, and that particular cavity starts with small one and becomes large one. So this particular, once this cavity starts appearing within this particular thecal cell or the granulosal cell, this primary becomes secondary. So then before the cavity, we call them as primary. Once the cavity starts in, we call them as secondary. Oocyte, it remains the same, but it gets the stimulation, it gets bigger, and it remains the same, is arrested there. Once this thing's cavity is full grown, it creates a cavity like this. You see that? A big cavity, and it filled up with a fluid. We call them as antral fluid, and this cavity we call them as antral cavity. Now once these things are created, surrounding this particular granulosa cells, you see that this is the ovum or oocyte that we are seeing here, and this oocyte is surrounded by a cell layer. So this particular cell layer which is surrounding here, we call them as corona radiata, which is surrounding it, and a portions of the corona radiata is anchored to this end, other end is free. So this point we call them as cumulus oophorus. So we have different names for this particular description part, doesn't have much of significance, but for descriptive reason we have to know what do you mean by this particular corona radiata. Corona radiata is single or multiple layers of this particular granulosa cells which is surrounding this developing oocyte, primary oocyte, and a layers which is anchoring to this particular granulosa cell, to this particular pole, anchoring to it. So we call them as cumulus cell or cumulus oophorus, and then it is filled up with this fluid over there. Now this particular secondary follicle, once this gets stimulation, the primary oocyte starts second cell division. So it becomes secondary oocyte and first polar body is projected. Now once this particular first polar body is produced and the primary oocyte completes the cell division, it becomes secondary oocyte. And because that becomes the mature follicle, we call them as Graafian follicle. So we get 4 stages: primordial follicle, primary follicle, then we get the secondary follicle, and then we get the mature follicle, we call them as Graafian follicle. The Graafian follicle is matured, full enlarged, just visible through naked eye, and this particular oocyte is different. In case of the primary, primordial and the secondary, it was primary. In case of the Graafian, it completes the cell division and it becomes secondary oocyte. At the secondary oocyte stage, this Graafian mature follicle is ready to eject this ovum out, surrounded by the corona radiata, Juna pellucida. They are thrown out. So this secondary oocyte is thrown out into the system, picked up by this fallopian tube. If the fertilization takes place, this secondary oocyte continues this particular maturation, which is arrested at this metaphase. If it is not, destroyed. Alright. So this is how these things are over. Now what happens to the follicle? It becomes degenerating follicles. The cells which is present, the theca interna, becomes filled up with some pigment cells. And once these pigment cells are produced, this theca interna cells can produce hormone now. The hormones which is produced by this particular theca cells are remaining for some times. If it is fertilization takes place, if the implantation takes place, then we have these continuations of this particular corpus luteum. We call them as corpus luteum of pregnancy. If it is not, this becomes corpus albicans. Okay. So what you have to understand? We have to understand this particular architecture of this element, how these things look like, cortex and medulla, and they have the different cells in maturing phase of the follicles are present: primary, primordial, secondary, Graafian, etc., and surrounding by different kind of granulosa cells, and depending on how the granulosa cells are layering, we name it accordingly. But point is that we have to understand this particular thing about oogonia or primary oocyte. So primary oocyte is arrested at prophase 1 till the puberty in the diplotin stage. Once the hormone sets in, we have the cell division gets in. Again, the cell division gets arrested at this particular meiosis continued. Graafian follicle, it becomes secondary oocyte, and this again it gets arrested at metaphase. It only gets completed the second meiosis if it is fertilization happening. If it is not happening, it is destroyed like this, secondary oocyte. So this is the oogenesis phase, and it starts with primary follicle, primordial follicle, secondary, tertiary, Graafian, and ovulation, eventually corpus luteum. So this is the different stages that we are going to see. How the cellularity is present, that we have to see. The difference between primary and secondary is having this particular pond inside. What do we call them as pond inside? Follicle, antral follicle, filled up with fluid. And then we have this particular Graafian follicle, which looks like the large follicle, secondary oocyte, cell division is taking place. These are the corona radiata surrounding this, some of the cells which is anchoring to this part, we call them as cumulus oophorus, and then this particular element is ready to be thrown out, ruptures, and secondary oocyte is liberated. The Graafian follicle is liberated with these particular cells. So it's secondary oocyte surrounded by the GP coat. Then we have this particular corona radiata. They start the venture migrating out into interstellar spaces. If we get this particular ovum met, then the secondary meiosis 2 takes place. If not, it is destroyed like this, phagocytes. So this is about different stages: primordial follicle, size, shape, etc., etc. The theca cells, theca interna, theca externa, which is modified granulosa cells, and then we have the antrum, which is the hallmark for the secondary follicle, and then Graafian follicle. The hallmark is the primary oocyte completes this fast meiotic division and becomes secondary oocyte. That's the thing. Second thing is that We have the corona radica GP, and this theca interna cells starts secreting the hormones, becomes a hormonal body. Then ovulation, the ovulation has a time, has a hormone sequence, has a sequence of this particular prostaglandin, which is basically making this particular things to act. Then we have this particular secondary meiosis arrested at this metaphase, remains there, only it gets fertilized, then it completes, and then it becomes the dead cell, that call them as corpus luteum. If pregnancy happens, we call them as corpus luteum of pregnancy, or else it is corpus luteum, it remains there, eventually disappears by the end of the female cycle, and we call them as corpus albicans, becomes scar tissue, no more hormone production, again new cycle begins. So, this is how this whole thing comes here. This is a Graafian mature follicle, large size, the largest of all, and this particular oocyte is in secondary oocyte. Corona radiata, cumulus superus, ZP, zona pellucida, glycoprotein coat, and then we have this antrum filled up with the antral fluid, theca interna, vascular layer, theca externa, then follicular layer most outside to it. Corpus luteum. Once these things are thrown out, then empty sac becomes only hormone producing element. So, you have to remember this particular element about this particular histology of the ovarian follicles at different stages. Clear? So, this thing we just remember, rest thing you can manage. So, this is the basic chart that we have to follow. Clear? So, we move to other histological structure, we call them as fallopian tube. So, once these things are ejected, we have this fallopian tube there extended, have got 4 parts, and we have this particular finger projections, we call them ephemerae, makes a negative current, and these things are sucked in by this fallopian tube. And this particular fallopian tube have got different parts, grossly we know this, but histologically if you cut this, gets into 3 layers. Most outside we have the serous layer, then we have the muscle layer, then we have the mucosal layer. The mucosa is extensive, it looks like a fern. You have seen fern, the planet, the primitive planet fern. The mucosa is highly convoluted, it looks like a fern. The convolutions of this particular mucosa having got epithelium layer, and it is resting on the basement membrane. The epithelium layer is ciliated epithelium, which is columnar variety, and it contains mucus secreting cells, ciliary cells and mucus secreting cells. We call them as PEG cells, which is basically fluid secreting cells, which is secreting the fluid inside this, and this PEG cell feeds this particular travelling ovum or embryo. So, this particular fallopian tube inside layer mucosa is most extensive. This is the hallmark to identify, they look like fern. Then we have the muscle layer, which have got smooth muscle, inner circular, outer longitudinal, and most outside the serous layer. Now, this is having the mixture of the things, mucosa, muscle and serous. The hallmark is the mucosa, which is having ciliated simple columnar epithelium and non-columnar epithelium cells, we call them as PEG cells. Now, how do they look like? This particular element, as you can see, this is outside layer. So, serous layer, this is muscle layer, outer longitudinal and inner circular. Then we have this extensive mucus layer, and mucus go on into folds. This particular folds looks like a fern. So, we call them as fern petal mucosal folds or epithelium. If you zoom this individual like this, they look like this. So, they look like villi, isn't that? So, that villi of the intestine, they look like this. So, this particular villi cells, they have this dot cell, brush like cells, which you see the ciliated cells. In between non-ciliated cells, we call them as secreted cells, PEG cells. They secrete lot of fluid inside the lumen for the nutrient rich substance to feed this particular developing or travelling ovum. So, this is how this whole thing look like. So, remember this particular picture, and it looks like likewise in the slides or microscopes. They look like this fern pattern mucosal folds. Remember this mucosal fold, highly convoluted and a thick muscle layer, which you cannot show, which you cannot see unless you move the slide. We show this particular fern pattern. So, looking at the fern pattern, you should be able to figure out this is a specimen of the uterine tube or fallopian tube. Clear? This is one of the easiest specimen, 2 of them we are learning. Moving from there, we have this reproductive system for the male system. Only one, which is easiest one, I would say, amongst these histology pictures. So, we have this testis to understand in here. Now, you see this testis which is placed in the scrotal sac having a covering. So, outer covering we call them as tunica, vaginals. It has got two layers, parietal layer, visceral layer. In between them we have a fluid collection, we call them as hydrocele. Then we have this white layer, albuginea. Then vasculosa, 3 layers. Now, this albuginea, this is splitting from this particular poles and splitting this particular into small lobules which is created in it, and within this particular lobules we have called superconvoluted tubules, we call them as seminiferous tubules. Now, the seminiferous tubules is coming from the same renal system tubules which is been unused by this kidney system, and this metanephric tubules mesonephric tubules are utilized by as seminiferous tubules up to this rated testis. So, seminiferous tubules gets into these tubules, we call them as rated testis. From here it becomes straight tubules, efferent ductules, then it becomes epididymis, have got parts head, body, tail, and then vas. This is how this whole thing is. Now, we cut this particular seminiferous tubules and get inside. So, this particular seminiferous tubules have got two kinds of cells. One is migratory cells, we call them as germ cells. They are coming from yolk sac, migrating in here. One is residential cells, the Sertoli cells. So, the feeder cells which feeds this particular this sperm cells, which is spermatogonial cells, which is coming in here, and this particular cells which is present, which is neighbor cells, we call them as Sertoli cells. And apart from that, outside the seminiferous tubules we have a cells, we call them as Leydig cells. So, these are the 3 different types of cells which are present. So, it is the inside this particular lumen is surrounded by epithelium, which is stratified epithelium, seminiferous epithelium, which is consisting of the spermatogonial cells, spermatogenic cells, migratory cells, and native cells, Sertoli cells. Sertoli cells are the cells which is maintaining the internal atmosphere within this particular lumen. It has got tons of functions. So, this is how this particular cross sections of the testis looks like: tunica parietalis or vaginalis, albuginea, and then we have this vasculosa which is inside to it. Then these are the seminiferous tubule cut sections. Within this cut section it has a lumen and it has adlumen, this side with the margin of this particular lumen. This is the central part of the lumen, and this is where you see that this is sitting on this particular cells which is germ cells. Within here there are some cells present as Sertoli cells. So, we have 2 cells present in here, germ cells and non-germ cells. Non-germ cells are Sertoli cells which is present inside to it, and the spermatogonial cells. Spermatogonial cells are of different types of cells. So, we have Sertoli cells and different phases of sperm cells or spermatogonial cells are there. They are known as spermatogonia, primitive one, type A, type B, type C. They are the most primitive one. Then from there we have the primary spermatocytes, secondary spermatocyte, then sperm teats, and eventually sperms which is present mostly in the epididymis region. And apart from that, this is the native cells. They looks like a cactus tree, right? Looks like a extended arms there, which is having a base and has a projected part, centrally placed nucleus or the basal placed nucleus. So, this particular part we call them as Sertoli cells. So, Sertoli cells are present within this part, and we have this sperm cells which are migrating in. So, this is a epithelium basal lamina which you see here. Into here we have this particular long cactus cells, we call them as Sertoli cells. Then we have this germ cells, spermatogonial cells, type A, type B. They are keep on migrating and moving up, and eventually the sperms of spermatozoa are present on the lumen side. So, if you go back to this particular picture, this is the lumen side, mid portions of it, which contains the sperm cells or more mature cells. Go to the base, we find more immature phases of spermatocells, and along with that we have the Sertoli cells. So, this is how the whole things are present. Now, the functions of the Sertoli cells physiologically that you have to know, it secretes numbers of factors. Most important thing is that they holds or secrete a protein substance, we call them as androgen binding protein, ABP. This androgen binding protein holds this testosterone inside. So, this particular internal environment of the sperm development is getting this particular vibes for the maturations from the like a spermatogonial cells, so that they become the sperm cells eventually by this particular presence of the testosterone hormone or dihydrotestosterone hormone. This is the sperm cells, and again this particular testosterones are produced by this element. We call them as Leydig cell. They are present outside this particular lumen. So, these particular lumens are there. This is the seminiferous tubules, 1, 2, 3. In between them, we have these particular cells which is present in between them. So, this particular place where these particular convolutions of this particular or conjunctions of these particular seminiferous tubules are there, these narrow gaps are there where you can find the Leydig cells along by these blood vessels as well. So, we call them as Leydig cells which is present in between them. We call them as interstitial Leydig cells. These Leydig cells, they produce this particular testosterone, and this particular testosterone producing cells, they are present in this particular region. If you have this particular testosterone damaged or destroyed, we have this particular no testosterone produced, then this particular sperm dies inside to it. So, this is how this particular histologically they look like. So, seminiferous tubules with this particular lumens, and within this particular lumens we have these two different types of cells are there. So, histologically they look like the same. So, we have 5 or 6 slides of different histological content are there. Some of them are ovary, some of them are fallopian tubes, some of them are testicles. So, go and see them and identify them and identify this particular the tissues, and that is it.